There are 40 trillion cells in your body. Roughly 3/4 of them are microorganisms. Today, you don't expect very much from them, but soon that's going to change. And I'm going to talk to you about two new technologies we're building here at Stanford in order to put the microbiome to work in order to treat and prevent disease. The first story is about how we're trying to build microbiomes from scratch, and I'm going to lead in by explaining where this work came from. And it started with a guy named Jospert Keller, who is a doctor in the Netherlands. And I'm going to tell you what Jospert was doing and why he was so frustrated. His expertise is in treating people who have very serious infections of the gut caused by a pathogen named Clostridium difficile. People would generally come to him when their infection couldn't be treated by anybody else. And the reason Jospert was so frustrated is that he had a new way of treating gut infections caused by Clostridium difficile, but nobody would believe him. Here's what Jospert would do. He would find a healthy person nearby and ask them for a stool sample. He would take this stool sample into the lab, put it in a blender with some salt water, and then infuse it into the patient with the infection. And almost every time, this would be curative. Somebody who had been in and out of the hospital for months or even over a year would be cured almost overnight. And Jospert was frustrated because nobody would believe that this transplantation of fecal material, of bacteria from one person to another, could be so effective at treating Clostridium difficile infection. And so he did the only thing he could do, which was to run a gold standard clinical trial, a randomized controlled trial to prove that this was effective. And the results were stunning. It turns out that 80 to 90% of the people who received a fecal transplant were cured of Clostridium difficile infection. These are people who could otherwise have died, whereas only 30% of people who were given the current best treatment, an antibiotic named vancomycin, were cured. In fact, the trial was so effective that he had to stop it in the middle because it would have been unethical to keep people in the control arm of the trial. This was astonishing to everybody, and it made front page news in newspapers all over the world because such a seemingly simple treatment was proven so effective at a disease that could kill people. We, and among many others, were quite impressed by this, but we wanted to take it in a different direction. What we saw from that clinical trial and from roughly 35 to 40,000 fecal transplants that have been performed to date since then, is that not only is this procedure highly effective at treating infections of the gut, but that it could go into a new direction. Because it was so safe, because adverse events from this process were so rare, and because you could transplant feces from almost any person into any other person, there's no need, as with blood donation, for a relationship between the donor and the recipient. And because the norm is that the bacteria that you transplant from one person set up shop in another person's gut almost every time, this opens all sorts of possibilities for diseases that are linked in some way to the bacteria that live in your gut. However, we think that the key to unlocking the power of this kind of treatment is to use not a fecal sample from a donor, but a concocted, built from scratch microbiome. We think that this is going to be the key that unlocks the ability to use microbiome transplant in diseases like inflammatory bowel disease and in cardiovascular disease. And I'm going to explain why over the next few slides. I'm going to use a second story to help illustrate the unexpected promise of using a procedure like this to treat disease. And this is a story about cardiovascular disease, and it centers around my friend and collaborator, a guy named Stan Hazen at the Cleveland Clinic. Stan's in charge of preventive cardiology, which means he sees many, many patients who are trying not to get cardiovascular disease. And so Stan became frustrated at the lack of good tools, really tests that he could perform in his patients to tell whether they were at risk of getting cardiovascular disease. So he did something that is seemingly straightforward. He decided he was going to measure all of the chemicals in blood that he had banked from thousands of his patients over the years to see whether the levels of any chemicals in the blood would predict your risk of getting cardiovascular disease. At the end of this long combing process, he came up with a chemical that has a very long name, trimethylamine N oxide, or TMAO, that's at the right side of the slide. whose level was very tightly correlated with the risk of getting cardiovascular disease. The big surprise to Stan and to everybody else is that this chemical does not come from people. It comes from bacteria that live in your gut. In fact, it comes exclusively from them. They nibble on two chemicals that are in food, in fact, are enriched in meat and eggs, named choline and carnitine, and turn them into an intermediate chemical named trimethylamine. And then in your liver, that chemical trimethylamine gets converted into TMAO. And that's what ended up in your bloodstream. And that's what Stan found was very highly correlated with your risk of getting cardiovascular disease. In fact, he did a large study to show that simply by measuring the level of trimethylamine N oxide, TMAO, in your bloodstream, he could predict with very high accuracy your risk of dying over the next 5 years. And so people have become interested in finding ways to lower the level of this chemical by targeting the microbiome itself. One of the clues about how this might be possible to do came from an interesting study that Stan did subsequent to this work, in which he took a group of vegans and vegetarians, people who do not eat meat, and he convinced them, for the sake of science, to eat a steak dinner. And what he showed was that if you fed an ordinary person, an omnivore like me, a steak dinner, then the level of TMAO in their bloodstream would spike. That's the top curve in the slide. Whereas if he fed a steak dinner to somebody who was a vegan, then they would not produce any TMAO from it, meaning that they were missing a bacterium in their gut that converts the choline and the carnitine in the steak to TMAO. The reason this was so exciting is because it proved that it's possible to construct a community of bacteria that's missing the bug that produces the toxic chemical. And if we could do that and transplant it into a normal human, then we would greatly lower their risk of getting cardiovascular disease. And so what we really need to be able to do is to construct a bacterial community that has the bugs we want in it and is missing the ones that we don't. Shown schematically here, if this is your gut today, you have several hundred bacterial species living in your gut, and when you swallow food, they get the first chew, and from that they produce a whole slew of hundreds of chemicals that make it into your bloodstream. Some of them are shown on the right side of the slide. The idea is that if we could build a new microbiome from scratch and transplant you with it, then we could eliminate the production of the chemicals that we don't want to end up in your bloodstream. And again, we think this is going to be a key that unlocks not just new treatments for cardiovascular disease, but also inflammatory bowel disease and other things like liver disease. So we set out to construct a synthetic microbiome, a built from scratch, completely defined microbiome. In order to do that, Alice Chang in my lab mixed together 104 bacterial species that we got from dozens of different labs and culture collections. And we did this without knowing whether they would play well together or whether we could get reproducible data from an experiment like this. It was, in many ways, an outlandish thing to do. But what I'll show you in a moment is that not only do they play well together, but they behave remarkably well. You can see that if you take the bacteria and mix them together, that's the blob on the left side of the slide, and then use them to colonize a mouse, then they self-organize into a bacterial community that looks very much like what you find in a human or in a conventionally colonized mouse in the wild. In fact, we also saw that the community that we put into the mice was incredibly stable over time, meaning that it had this property of stability that emerges when a community of organisms is sufficiently complex. And so we had built something that was sufficiently stable and looked enough like the real thing that we think this is going to be the basis for a lot of therapeutics moving forward. We then showed, in work that Min Wang in the lab did, that we could remove a single organism from the microbiome, something that people have dreamed of doing for years but have not been able to do because it's difficult to take an undefined sample from a person and take one bug out of it. But we, since we know all of the parts in this bacterial community, can now remove just one and see what happens. And when we colonize one group of mice with the complete microbiome and one group of mice with the microbiome that's missing a single bacterial species, we saw to our delight that almost everything chemically was the same about the two groups of mice, except for a chemical that we had hoped to eliminate in one group of mice, which is the chemical shown in the upper right-hand corner of the slide, which is also represented by the red bars, next to which there are no blue bars, indicating that we had, by eliminating one bacterium from the gut microbiome, we had gotten rid of completely the production of a chemical that we had hoped to eliminate. So we think that this technology is going to make possible a number of microbiome-targeted treatments for diseases that are not currently accessible. So that's one way that we're putting the microbiome to work in order to treat diseases that currently can't be addressed. I'm going to tell you about a second technology for the rest of the talk that has nothing to do with the first one. It's a completely different trajectory, and it is on the topic of vaccines, though I'll come back to that at the end. This is a shorter story, and to lead into it, I'm going to tell you something that seems ironic because we're in the middle of a pandemic, so this is an odd time for me to remind you that most of the time, most people are not sick. That doesn't mean that your immune system is off. In fact, most of the interactions that your immune system has under normal circumstances are with the bacteria that line all of your barrier surfaces. That's your skin, your mouth, and the lining of your gut. This is a very rich environment for immune cells that are not ignoring but constantly interacting with the organisms from the microbiome that live in close contact with you. And the story today is going to be about how we're going to engineer an organism that lives on the skin in order to do something useful for our immune system. There is an observation that people working on the microbiome have made over the last few years, which is very simply that certain organisms that live in the gut and on the skin induce a very potent immune response with no symptoms in an animal. So if you colonize mice with the pink bacterium or the green one or the blue one, you get very sharp immune responses that look very much like what would happen if you had gotten infected. And yet the mouse looks perfectly healthy. So this brings to light the possibility that the immune system is surveilling the organisms that live on and inside of an animal, even of humans, and responding to them actively in a way that you would never know if you were just walking around. We wanted to take advantage of this and see whether we could redirect that immune response against a target that we would all like to treat, which is cancer. So we decided to take the following approach. We started with a bacterium that lives happily on your skin named Staphylococcus epidermidis. Every single human on the planet is colonized with Staph epidermidis, and certain strains of this bacterium, when they colonize your skin, induce an immune response that looks very much like the immune response you would want to attack a tumor. It is a kind of immune cell named the CD8 T cell, and this is exactly the kind of immune cell that you would want attacking a tumor. But of course, the T cells that are induced by the bacterium living on your skin are looking for the bacterium living on your skin, so they're not very useful to us right now. What Erin Chen in the lab did was to put a little piece of the tumor inside the bacterium. She made it look on its outside like it was molecularly the same as the tumor. And then she colonized mice with this bacterium that had a little piece of the tumor in it. And what she was hoping to do was to trick those immune cells into not looking for the bug, but looking for the tumor and killing it. And so what she did was to put tumor cells into a mouse and then take a Q-tip into a culture of this engineered bacterium and rub it gently on the head of the mouse. And what she was looking for was the size of the tumor to shrink. She found exactly that. If you don't put any bacteria onto the mouse, the tumor grows very rapidly and kills the mouse quickly. If you put the normal bacterial isolate onto the mouse, exactly the same thing happens. There's no immune response induced that has anything to do with the tumor. But if you put a little piece of the tumor into the bacterium and then colonize the mice, that's the blue dots on the right side of the slide, then you see the size of the tumor shrinks dramatically. This is something that's very exciting to us. What's even more exciting to us is that it seems to work even in the context of the most difficult cancer that we could give the mice, which is a metastatic melanoma, a cancer in which the tumor cells set up metastases in the lungs of the mice. And even under those circumstances, just gently swabbing this bacterium onto the head of the mouse made those tumors shrink dramatically. We think that this is going to open new possibilities in treating tumors because we think what we're really doing is from a distance on the skin, we're inducing tumor-fighting immune cells, except by engineering bacteria that live on the skin to look a little bit like the tumor. Then those immune cells leave the skin, go find the tumor, and then blow up the tumor cells, causing the size of the tumor to shrink without causing any other side effects in the animals. We're very excited about seeing this through to the clinic, and that That program, along with some of the work that I told you about earlier in the presentation, is what we're focusing on in the Stanford Microbiome Therapies Initiative. Thank you.